finish lecture 2 today. And so, last time, if I recall right, we were on this slide and going through the characteristics of skeletal muscle. So I thought I'd kind of review that with this. Skeletal muscle cells are long cylinders. So when you're thinking about that in the lab, and you're looking at the muscles and how they've kind of drawn the pattern of fibers on the muscles, then just keep in mind that the cells are running from the tendon of origin to the tendon of insertion. So these are actually really long cells relative to what we kind of talked about in the past. And they're actually a fusion of cells from embryonic development that retain nuclei. So they're an example of cells in our body that are multinucleated. We just can't do it. Bring on the So they're multinucleated, which is kind of unique in the human body itself as well, in terms of cells with more than one nucleus. Because they do form from embryonic fusion, there's a repeated pattern to them. And so they appear to have stripes on them that we call striations. So they're striated. And what we use is the word sarcomere to delineate that pattern. So remember that it's a repeating pattern of dark bands and light bands. So on Tuesday, we said the dark bands were A bands, yeah, and the light bands are high bands. So it's a repeating pattern. What we're going to talk about today is how calcium is so critical to the ability for muscles to contract. And so if you go get your blood tested and they test for the electrolytes in your blood, you would see that we don't carry a lot of calcium in our blood. In fact, we store most of our calcium in our bones. Yeah. So for skeletal muscle to work, they also have to store calcium. But they can't store it the way bone stores it, or we would have crystalline structures within skeletal muscle, and then the muscle wouldn't be able to contract very well. There's actually a rare genetic disorder where people begin to ossify their muscles, and then they lose contractility in the muscles. And it's because we store a lot of calcium in the sarcoplasmic reticulum. And so again, endoplasmic reticulum that we've talked about in the past, we call rough if it was involved in protein synthesis. We call it smooth if it was involved in lipid synthesis. So we use sarcoplasmic instead of endoplasmic to describe it as unique to muscle tissue, and then it uniquely stores calcium. And then in a little while we're going to talk about what the role of that calcium is. And then we talked about the fact that these cells are really giant cells, and one of the things that constrains the ability to move things in and out of the cell is surface to volume ratio. So throughout our body, we see unique ways of managing surface to volume ratios. So for example, when we did columnar cells, we talked about folding membranes on columnar cells called microvilli that increase surface area. Those were upward folds. What we're going to see in skeletal muscle is downward folds of the membrane into the cell. And those downward folds are called transverse tubules. And they're going to help increase surface to volume ratio on the cells themselves. And then what we were just getting to at the end of class was the fact that we have these junctions that allow cells to actually communicate with one another. If the cells are held tightly together. And there are actually little proteins called connectons that actually connect the cytoplasm of one cell to a neighboring cell. And so that allows for the cytoplasm over here to actually be exchanged with this cell. And so they're communication junctions, so they're called gap junctions. And so in skeletal muscle, we don't have So there are no gap junctions in skeletal muscle cells. So each cell is completely separated from neighboring cells. Then to reinforce that, if we go back and think about the hierarchical layer of connective tissue that we talked about on Tuesday, then the connective tissue that surrounds the outside of a muscle and separates muscles from each other was the epimysium, remember? And then groups of cells form subunits within a muscle we call fascicles, and it was surrounded by the perimysium. And then each individual cell was surrounded by a connecting tissue layer we call the endomysium. So the endomysium further isolates cells from one another. So the cells really do not communicate with each other at all. So that creates a problem in the brain's ability to control skeletal muscle activity. And so when we look at that, what we do know is that skeletal muscle is not autorhythmic, meaning that it will not contract without your brain sending a signal. Okay. So we see this clinically where somebody's had spinal cord injury, and where the spinal cord was injured, the information coming down from the brain can no longer go out toward the muscle, and then the people are paralyzed because the brain can no longer control the muscle. You could also see it in people that have had strokes, where actually a part of the brain was damaged, and if that part of the brain was actually controlling skeletal muscle, then they also have paralysis. So we've got this strong connection between our brain and skeletal muscle to create activity. And so we say that is voluntarily controlled, meaning that you physically have to create the thought process with your brain to actually be able to do that. So we're going to put this together in the next unit. But when you kind of look at the brain, this is why I do PowerPoints. But this would be our frontal lobe. And the back portion of our frontal lobe is critical to controlling skeletal muscle. So what we have is we have cells that are in the frontal lobe of our brain, and they actually have to send signals down that would go out to our body via nerves that would control your skeletal muscle. So we originate the contraction up here, the information that's going to cause the contraction. Then we have to send it down our spinal cord and out to a muscle. So that's why if we damage our spinal cord, so that this can no longer communicate, it's actually going to broken, then we'd have paralysis. And it's also why if we damage this area of the brain, where this no longer functions, why we would have paralysis. Because that part of the brain is critical to skeletal muscle contraction. And every time you try to do something, you have stored memory based upon the sequential contractions required to make a task. So you weren't born knowing how to tie your shoes. You had to practice that. You weren't born being able to throw a basketball or a football well. You had to practice that. So there's a part of your brain that has retained the memory of the muscles required to accomplish that task. So some people, when they have strokes, will not remember how to tie their shoes, or can't remember how to write. And what we have to do is retrain the brain for that task again. And that's what OTs and PTs spend a lot of time doing with people, particularly OTs, is training them to accomplish tasks that you and I take for granted.
So what would cause temporary paralysis?
There could be a number of things. One of the things is a couple of viruses will cause temporary paralysis. So there's a whole host of things that could do that. From electrolyte imbalances to bacterial viruses. Right. So our muscles just don't do their own thing. They don't have that capacity. They require a brain, and our brain has to send the signals. That's why we call it voluntary. Skeletal muscle has got an incredible speed of contraction. It's the fastest of all of our muscles in the ability to create a contraction. And then what we're going to do is actually talk about next week about how muscles are constructed with different types of cells, and the cells themselves have different speeds of contraction. So that our hands have very rapid contractibility compared to the legs, and so we'll talk about that. So not only does skeletal muscle have an incredible speed of contraction, but we have cells that vary in that speed of contraction. And then muscles are constructed with different ratios of those cells that provide different functionality for muscles. Okay. And so, because there are no gap junctions between muscle cells, and because our frontal cortex is critical to controlling skeletal muscle activity, then we have to have a way in which our nervous system communicates with skeletal muscle cells. Right? So every cell, every skeletal muscle cell, actually has a connection to a nerve that would be coming from a nerve in our central nervous system. And the nerve is not actually physically in contact with the muscle cell itself. So we actually have a little space. And because we have a space, then the nerve has to release a compound that's going to cross the space and go to the muscle cell itself. So what we do is we call the compounds that cross this space generically neurotransmitters. And that's because they diffuse across the space from where they're being released from the nerve at high concentrations to the membrane of the skeletal muscle cell, which is in low concentrations. So the neurotransmitter that's critically important to skeletal muscle activity is this compound called acetylcholine. And so acetylcholine is our neurotransmitter that communicates with skeletal muscle. So in World War II, we actually had figured this out and began to realize that if you could interrupt these neurotransmitters, then you could interrupt skeletal muscle activity. And so one of our first chemical warfare agents was a compound that actually disrupted the process that acetylcholine was involved in. And it actually blocked an enzyme that breaks down acetylcholine. Because if you want a muscle to relax, you've got to remove the acetylcholine. And so we have an enzyme that breaks down acetylcholine. The nerve gas, or chemical warfare agent, actually disrupted that enzyme so that acetylcholine would stay bound with your skeletal muscle cell. And when it did, your body went all stiff and your muscles all stayed contracted. And you could not relax. That would include your heart. It stayed contracted and not relax. So it killed people very effectively. And they couldn't shoot back at you because it couldn't actually move you. Yeah. So, and it just actually interfered with this process of communication with the neurotransmitter. And then the interesting thing about skeletal muscle cells is that they have limited regeneration. So we know that there are these stem cells that we find interspersed through skeletal muscle cells that are called satellite cells. And they're committed unipotent stem cells that will allow for limited regeneration. There's been some research using stem cells to see if we can trick them into becoming cardiac muscle cells as well. So that's one of the areas of research that may allow us to actually repair heart. All right, so let's kind of look at what this really looks like. Let's look at my crude drawings. Okay, so the skeletal muscle cells themselves are quite long. And what we're going to see is the word sarco used in several ways, all unique within a muscle. So we talked about the sarcoplasmic reticulum. We talked about the subunit, the sarcomere, and the membrane surrounding a muscle is referred to as the sarcolemma. So the plasma membrane, because of its overlying connective tissue layer, the endomysium, is referred to as sarcolemma. So word lemma means layered, sarco meaning to muscle. So it's a way of telling you that the plasma membrane of a muscle is a layered structure. So it's surrounded by the sarcolemma. And then we have a number of nuclei. And because the center of the cell is going to be dominated by the contractile proteins that allow a cell to contract, then skeletal muscle cells, like fat cells that we talked about in the first unit, don't have their nucleus at the center of the cell, but the nuclei are clear pushed to the edge of the cell. And so these are the nuclei right at the edge of the cell. And the inner part of the cell is actually filled with plasma proteins that allow the muscle to contract itself. And so what we have then, because these cells are big cylinders, is we have these downward folds of the plasma membrane. So the plasma membrane is kind of blue here. These downward folds are blue because they're trying to show you that it's part of the plasma membrane folding downward. And that was the T-tubules that we talked about, that increased surface area. And then when we look at the way that the sarcoplasmic reticulum is found within a skeletal muscle cell, it's not a random organization, but actually a highly specific organization, so that large blocks of the sarcoplasmic reticulum actually line up along T-tubules. So we end up with large blocks of the sarcoplasmic reticulum surrounding the T-tubules. And so these large blocks are called terminal cisterns. And so a cistern is a storage vessel by definition. So these are called cisterns because they store calcium. So that's actually where the calcium is being stored, in these terminal cisterns. And then what happens is that we have two specific types of proteins that have to interact to create a muscle contraction. So they have a generic name we're going to use first, and then as we talk about the process, we're going to go to a more specific name. So oftentimes when you see a picture of a muscle cell like this, they have thick filaments and thin filaments. And thick filaments and thin filaments are actually the proteins that are required for contraction. And they get their name because one of them is large, so it's thick. One of them is small, so it's thin in terms of their diameter. Okay? And what happens is the thick and thin filaments interact with one another to make up this kind of tubular-shaped entity that's within the cell. And so it's called a myofibril. Okay? So the way to think about that is you've got this long cylinder cell, and we could call that a fiber. So we would see it in text commonly described as a muscle fiber. Okay. So then the muscle fiber itself is made up of these much smaller long cylinders created by these contractile proteins. And so it's a repeated pattern of these long cylinders. And so since the large entity is a fiber, then the subunit under it is a fibril. So if you have a fiber and you have smaller subunits of fibrils. So the way to think about it is if you had a rope and you called the rope a fiber, then all of the strands that make up the rope would be fibrils. That's the way it would.
Would that be considered an organelle?
They're not technically organelles because they're actually constructed of proteins. And, all right. So then the other thing to remember is that the word muscle, we can use the prefix myo. So if we want to be more technical about a muscle cell, we can say it's a myofiber. And then the subunit becomes a myofibril. So that's where the word myofiber is coming from. It's actually telling you muscle fiber. Okay. And then as I indicated, the word sarco, unique to muscle, lemma layer. So it's really the cytoplasmic membrane plus its overlying Myosin. That's above. So the story of muscle is really kind of cool. And it's kind of a fun way to think about how science really works. And how discoveries take the talents of a number of different researchers, sometimes from different disciplines, which is why usually if you're going to go into biology, you have to have a little chemistry and you have to have a little physics to go with biology. Because oftentimes it's been the collective work of biologists working with chemists or biologists working with physics, physicists or physicists and chemists and biologists working together that have actually been able to create a lot of the knowledge base that we take for granted. So remember Watson and Crick, who elaborated the structure of DNA. One was a biologist, one was a physicist, working together to kind of work on that. So what the biologists have typically done is take the material from the tissues from animals and try to explore how they're put together in relationship to visual imagery and stuff. Okay, so when you look at a muscle under the microscope, and I'm just going to dim the lights. So what we see is this repeated pattern that we just talked about: dark band, light band, dark band, light band, dark band, light band, and then here are the nuclei, sandwiched clear to the edge. And so when we first looked at that, we eventually called the bandings A bands and I bands. And then the question that arose is, why do they exist? Why does this repeated banding pattern exist? What internally within a muscle cell accounts for that? Okay. And that's where biochemists came in. Because biochemists like to take cells and grind them up in blenders and homogenize them and then chemically analyze the stuff that's inside the cells. So when I was a graduate student, it was terrible. The story was if you give a biologist a frog, they'll watch it, they'll explore it, they'll dissect it. If you give a chemist a frog, they put it in the blender and turn it on. Sorry, chemist. Well, what really helped us was as we moved from light scopes, which had limited resolution, to electron microscopes that gave us much more detailed resolution. So to put that in context, I was in graduate school in the 70s, and when I first started in graduate school, the biology program that I was a graduate student in got its first electron microscope, and so that they could actually use it. So when we invented an electron microscope, we were actually now able to see a little more detail to begin to try to figure out how in the world these striations were kind of constructed. And we had some knowledge from the chemists that would begin to tell us what those thin and thick filaments were constructed from. So we had this kind of chemistry basis. And so with the electron scope, what we were able to see that we couldn't see in the last slide was the fact that there's actually a dark line at the center of every I band. And there's a light band in the center of every A band. So that instead of just being an A band, I band repeated pattern, there was actually subtle detail within it. So that we had this dark line here and this light area here. So because we already kind of understood, because of the biochemist's work, what the thin filaments were constructed of and thick filaments were constructed of, it allowed us to kind of think about the pattern that we were seeing within the cell itself. So what did the chemists tell us? Well, the chemists actually told us that the thick filaments were actually made up of a protein, complex protein molecule called the myosin molecule. And again, So the cool thing is if you discover a molecule, you get to name it. So since this was unique to muscle, they put the prefix myo in front of it. So myosin was a protein that appeared to be pretty unique to muscle cells themselves. And it kind of looked like golf putters. And then the golf putters are all constructed into a thick filament that's made up of repeated myosin molecules. And so to kind of think about that, then what we had in an A band was we had that kind of light area at the center of the A band, and then we had that kind of dark area at each edge. Well, once they understood how myosin molecules were constructed, it gave them insight into why we would see a pattern like that within an A band. So the first thing you have to understand is the A band is where myosin is. And then the second thing is these heads would stain darker than these tails. And so since the heads are all pointed outward from the center of the molecule, then this part of the molecule would stain darker than this part of the molecule. And so once they figured that out, it was like, wow, this makes sense. Why am I seeing this lighter piece here in my A band? Because the myosin heads are all pointed to the outside edge, and the myosin tails are toward the center. And it's just a matter of how much protein material you have to absorb stain. And so it actually stains it. Isn't that cool? Yeah. Right. So then they also figured out there was this other protein within muscle cells themselves. And it actually was a protein constructed differently, and then it had another set of proteins that it interacted with. So the protein they figured out first was this one, which is kind of a globular protein, and then it's put together end to end like a pearl necklace. And that these thin filaments were actually 3 long strands of actin winding around each other, like 3 pearl necklaces. Okay. And so they were called actin because we eventually figured out that they were the protein that was actually going to move or glide to create a muscle contraction. So they're the active protein, hence the name actin. Okay. And then the other part was that we had these 2 other proteins that interacted with it. So tropomyosin and troponin. And then one other piece that took a little longer to discover. So these are all pieces of research that are occurring over time. And then the researchers have the opportunity then to read everything that people have been doing and then to begin to construct knowledge out of all these fragmented pieces of research. So they also figured out that for a muscle to work, myosin has to attach to actin. And so these little brown things that are shown here is actually where actin and myosin are going to interact. And then we're going to come back to that piece. So if you know what the actin looks like, you know what the myosin looks like, and you have this visual image of what a muscle cell looks like in terms of a banding pattern, then what you do is you just begin to construct things. So what's amazing is that Watson and Crick created the first model of DNA by using toothpicks and gluing them together. So think about that for a minute, because that was always just fascinated me. Because if they won a Nobel Prize by looking at a bunch of work that had been done and thinking about images they'd seen and constructing a model out of toothpicks.
I think kind of their competitors' research as well, but
there's some little stories like that. Yeah. Science tends to be ruthless like other areas of societies. And so this is so the last picture I showed you was a scanning electron micrograph. They gave you the 3-dimensional perspective of the cell itself. This is actually an electron micrograph that gives you incredible resolution but it doesn't give you three-dimensionality. So this is an A band, and this is an I band. And so when we saw that, and saw this linear relationship right here, and this linear relationship right here, that's what actually told us how actin and myosin actually fit together. And the myosin molecules are found here, and the myosin tails are here, and the myosin heads are here. And these are very thick in appearance, and these are very thin in appearance. So these are the actin molecules out here. So actin out here and myosin here. And then, even though there are myosin heads here, it's lighter here than here, but really light at the center. So I told them that the tails were actually at the center, and that we have myosin heads that are not overlapping with actin, and then we have actin and myosin overlapping. So that explains why this is so dark right here, and right here was the fact that we have this actin and myosin overlapping. Okay. So then once you do that, you create this little toothpick model. Then what you do is you begin to name the components that you actually see. So this dark line became known as the Z line. And it actually became known as the Z line because it actually does, under more fine resolution, actually Z back and forth. Okay. And this is what organizes the actin. So the Z line is what organizes the actin molecule. And that's in an I band. So then in an I band, we have this area that doesn't stain dark right here. This area right here. So that's been called the H zone. And then we have this overlapping thin and thick filaments to either side of that. And so the other thing that they needed to try to figure out was why in the H zone we actually have another dark line. So even though this is lighter through here, we actually have another dark line in the middle of the H zone. So the amazing thing about computers is that computers let us model all this stuff to look at three-dimensional things and look at how they could all work together. So the beauty of today's world is we have these amazing ways to three-dimensionally model things. So when you put what we just saw as kind of a planar, non-three-dimensional view, you begin to construct a three-dimensional model out of it. Then what you begin to see how it all fits together. So here's the Z line, because it actually does Z back and forth. And there's called a Z disc where the actin molecules are attached. So this line organizes our actin molecules. That dark band at the center of the H zone was eventually elaborated, and it's called the M line. And that's what organizes the myosin molecules. So we have these two lines that organize the molecules three-dimensionally. The Z line organizes actin molecules, and the M line organizes myosin molecules. So M line because it organizes myosin molecules. Okay. All right. And then we have the H zone here, where the tails are. And then we have this zone of overlap where it stains dark. And so now when we look at an A band and an I band, we've been able now to kind of look at how this was constructed embryologically. And so now here's our sarcomere, which was the unit we talked about that created the striation. And a sarcomere runs from the Z line in one I band all the way to the Z line in the next I band. So a sarcomere is essentially half of an I band, all of an A band, and half of an I band. All right, so we're going to leave it there for a little bit, and we're going to come back and talk about how that thing works. It's pretty darn cool. Yeah. All right, so if we contrast skeletal muscle that has long cylinders, then actually that'll begin to make sense when you think about how the muscle works, because it's inserting on an origin, it's crossing a joint typically, and inserting on another bone that we call the insertion, and then the muscle is planarly contracting between those 2 points. Your heart is three-dimensionally round, and so what we have to do is we have to have cells that can contract on a three-dimensional plane instead of a one-dimensional plane. Okay. So long, straight cylinders work great if you're contracting on a single plane. If you're going to contract on more than one plane, long cylinders don't work at all. And so what we find with cardiac muscle is they're branching cylinders. So we go from these long cylinders to actually a cell that actually can branch. And then it can interact with more than one cell at the other end, which allows for more three-dimensional contractility. So they're branching fibers. And they aren't near as big as these giant skeletal muscles because they aren't spanning such distances. So they also only typically have single nucleus per cell. And it tends to be more like we see in most cells, more internally organized nucleus. Although if you looked at a bunch of myocardial tissue under a microscope, you could occasionally see cells with two nuclei. So it isn't uncommon to have binucleated cells, but we don't have multinucleated cells. Okay. So where the cells join, so the way to think about this is you can try to join a cell like this, or you could try to join as two cells like this. So intuitively, which would be the strongest connection? The bottom one, right? Because it all goes back to surface area. So if you can increase the surface area of contact, then it's going to be a stronger. So because the membranes actually overlap like this, they stain darker than the rest of the membrane material. So they've been called intercalated discs because of the staining pattern that occurs in it. So you get this dark band here where they're overlapping, you get a dark band here where it's overlapping. And they have very subtle A and I bands, but these really stand out. So they're called intercalated discs. And then there are gap junctions in these intercalated discs. So we do have a way in which one neuron, one cell can interact with a neighboring cell. So since these cells were all isolated, every muscle cell has to have a nerve that goes to it. Okay. If you can have gap junctions, then you can have a neuromuscular junction on one cell, but you don't need it on neighboring cells. And so by having gap junctions, each cell within cardiac muscle doesn't need to have its own connection to the nervous system. All right. So there's striated composed of sarcomeres again. So we do see A and I bands, but very subtle A and I bands compared to compared to skeletal muscle. And in some sarcoplasmic reticulum or calcium storage. But what we know about cardiac muscle is that if you had a patient whose blood calcium level was low, they would have cardiac dysfunction before they would have skeletal muscle dysfunction. And it's because cardiac muscle doesn't store as much calcium, and it's really dependent upon blood calcium levels. So if you actually looked at patients who had too low of calcium, you actually begin to see cardiac dysfunction because of it, way before you see skeletal muscle dysfunction. So we do have transverse tubules again, because these are still relatively large cells relative to all the cells we've looked at in the past. And so we do have to have transverse tubules again. And then what's amazing about cardiac muscle is that the cells have autorhythmicity. So this is really cool. I tried to find this video out on YouTube, and I couldn't find it again. But they've been trying to culture cardiac muscle cells, obviously, because if you can actually culture the cells in a petri dish, then you can actually then progress toward repairing damaged hearts, because you'd have to culture the cells. So they've actually been able to culture cardiac muscle cells in petri dishes, so we can actually do that. And so that what the video was two different petri dishes with cardiac muscle cells in it. The cardiac muscle cells were just contracting and relaxing in these petri dishes. And then what the researcher did is pour the contents of the two petri dishes together. So they were actually contracting at different rates. And when the cells came in contact with each other, they synced and contracted together. And yeah, because of this ability to intercommunicate. So pretty amazing.
How would they contracting and nervous system stick on?
Well, actually, the myocardial cells themselves have autorhythmicity. Your heart has a pacemaker that controls the autorhythmicity of the heart. You can actually extract a heart and it doesn't need the brain to beat. I used to do this with a frog when we did cardiac muscle. When we actually had where I talked before, where we had an animal facility where we could raise animals. So what I would do is I would take the frog, anesthetize it, cut its heart out, put the heart in the beaker. And as long as I kept the oxygen level in the beaker high and kept changing the water every once in a while to maintain glucose levels, I could keep the heart beating in the beaker for 6 hours through my labs, and it would just sit there. So our heart itself has an internal pacemaker that establishes the base rate of your heart. And what we need our brain for is to accelerate the heart rate when your body is under load, like exercise, and then bring the heart rate back to that base level when you no longer need that increased cardiac output. So our brain alters the base function of the heart. Neurotransmitters. Yeah.
So a heart on its own beats at its minimum rate and it can't beat any slower? Or
Well, you can make it beat slower.
okay.
But yeah, essentially establishes its own rate. And then it's influenced by neurotransmitters or availability of oxygen and nutrients. Okay. So it's quite interesting. So your heart is also involuntary, which is a good thing, because if you had to think about it all the time, then what would happen if you got preoccupied with a movie? Like, oh darn. So the heart itself, at a base level, doesn't need your brain at all, because it's autorhythmic and it's involuntary control. And it's a moderate speed of contraction. So as you begin to exercise and you're using more skeletal muscle as fast contracting, like in a sprint, then your heart accelerates its contractibility as well. Okay, but it's never as fast as skeletal muscle itself. And then here's the key, which is of what we've just been talking about, which is pretty fascinating, is that the heart creates its own inherent system. And our nervous system has to override that to accelerate the heart and to decelerate the heart. So if you have a task to do of increasing the activity and decreasing the activity, you can't use one neurotransmitter to do both. So you have to have a neurotransmitter that would speed the heart up, and you have to have a neurotransmitter that would slow the heart down. And so the nerves going to the heart have to release different neurotransmitters to actually manage that. Long story short, we'll actually in the next unit talked about the fact that the heart's innervated by two different branches of a system that controls most of your involuntary activity of your body, called the Autonomic Nervous System. So we have our buddy acetylcholine, which is exactly like the acetylcholine that we talked about in skeletal muscle. But acetylcholine actually depresses your heart rate. So that was actually the beauty of the nerve gas that they created in World War II. When you prevented acetylcholine from being broken down by an enzyme, acetylcholine load on your heart elevated and your heart rate plummets. And it essentially puts you in anaphylactic shock and kills you. Yeah, and that's the way it actually worked. So an example of that is what I just said. If somebody enters anaphylactic shock, then their heart rate begins to drop. Okay? So the hormone or neurotransmitter that actually accelerates heart rate is norepinephrine. And then there's a sister molecule that your body makes too, if you just take the nor off of it, would be epinephrine. So since anaphylactic shock puts you into a declining heart rate that eventually can kill you if you have really bad anaphylactic shock events, then most people who have an allergy that leads them into anaphylactic shock carry EpiPens. And the reason why they carry EpiPens is because epinephrine will convert your heart from dropping lower and lower and lower and lower, and it causes your heart to accelerate. And it keeps you from dying from anaphylactic shock. Well, people who were in Iraq, at least the first time, and maybe to some extent the second time, actually, because we knew that the Iraqi government had this chemical that from World War II, and we knew that because we had sold it to them, that soldiers carried EpiPens, and they had little detectors that detected the deal, and then they could auto-inject themselves with the EpiPen, and it would counter the effect of the nerve gas that was going to drop their heart. Yeah, so interesting. So anyway, so this decelerates your heart rate, and this increases your heart rate. So when you begin to exercise, then your nervous system releases norepinephrine. That accelerates your heart rate based upon the body's need for cardiac output. When you're done exercising, then acetylcholine is released, and it brings your heart back down to normal. So when I was a student taking a physiology class, and we were learning heart physiology, the guy would get cats and dogs from the Humane Society that were going to be put to sleep that day. And we actually did open heart surgeries on the cats and dogs so we could learn different aspects of the heart. And the way we killed them was to just overload the heart with acetylcholine. I mean, they were under anesthetic. It wasn't an experiment where they were in pain. So it was interesting. Now, the dilemma we have with cardiac muscle is no regeneration. And so if we could resolve that, the world would be very different for people who have heart attacks.
So with the nerve gas, if they took the EpiPen, wouldn't the rest of their muscles still be contracting?
No, because yes, but there's another antidote. But what you want to do is you want to prevent the heart from plummeting. So there was another compound that they carried called atropine. And atropine counteracted the effect of this on the enzyme. Okay, so when we look at cardiac muscle, we see patterns that we see in skeletal muscle, although much more subtle. And so you really have to look close to see the light and dark banding patterns throughout this, where the A bands and I bands are. And that's because these cells aren't as big in diameter, and so there's not as many thin and thick filaments in it, so it doesn't band as well. But you can see that kind of pattern here. Then what we do see is these really dark Okay, and those are the intercalated discs. Those are the boundaries of one cell and neighboring cells. And they're actually branching cells, so it's really kind of cool. So if you look at this cell right here, and here's the nucleus of the cell. Here's an intercalated disc connecting this cell to this cell. Here's an intercalated disc connecting this cell to this cell. Here's an intercalated disc connecting this cell to this cell. And here's an intercalated disc connecting this cell back to this cell. And so this cell is actually connecting to a number of different cells simultaneously. And that's what helps it create a three-dimensional capacity to contract. Okay. So it's really kind of cool the way it's constructed. And we can pick out these intercalated discs. And if you get a really good slide, you can just kind of do that, which is really neat to see how the cell actually is contracted. Okay. And then this is an electron micrograph, which allows us to see a little more detail. So now the A and I banding patterns are a little more obvious here. Dark band, dark band, dark band. Intercalated disc, and then the nucleus that we can actually see. And another intercalated disc up there. So now we want to go to smooth muscle. And smooth muscle is constructed very different than the other two. So what we're going to see if we compare the characteristics of the muscle is that cardiac and skeletal muscles share more characteristics than smooth muscle does with any of the other two. So smooth muscle cells are our smallest muscle cells, and they're spindle-shaped, so that they actually taper at an edge. And they actually have only one central nucleus that is much more obvious than what we saw in cardiac muscle. So spindle-shaped cells with a single nucleus. And there are no A and I bands. And so if there are no A and I bands, then that means that thin and thick filaments aren't constructed the same way. Because what created the A and I band was the organization of thin and thick filaments. So they don't have sarcolemma. So they don't have I bands or A bands. So they don't have this cool structure from one Z line to the next Z line. So they contract very, very differently than cardiac and skeletal muscle. They have the smallest amount of sarcoplasmic reticulum. So these cells are loaded with sarcoplasmic reticulum. This has some sarcoplasmic reticulum. And oftentimes you don't even see it in smooth muscle. So very scant sarcoplasmic reticulum. You could, if we wanted to simplify that, we could just simply say lack sarcoplasmic reticulum, because you don't oftentimes see it. Because the cells are so darn small compared to cardiac muscle and skeletal muscle, surface to volume is not an issue. In fact, because the cells are elongated and tapering, they actually now have a much higher surface area to volume. And so we don't need T-tubules anymore either, because that was unique to a cell that was big in diameter that had this surface issue to deal with it. So no transverse tubules because they're dinky compared to the other two. And then it varies. So like associated with your digestive system, where the cells overlap, they have gap junctions. So the ones associated with visceral organs like your small intestine, large intestine, stomach, actually have gap junctions. But the smooth muscle that makes the erector pili muscle that stands our hair up, lack gap junctions. So they live in both worlds. So some of it's like cardiac muscle where there are gap junctions. Other smooth muscle we find in the body is more like skeletal muscle where they lack gap junctions. So it's kind of a combination within the smooth muscle. And smooth muscle is autorhythmic. It doesn't need your brain. So there's always these life experiences that you have. And then when you learn something in college, it's like the light bulb goes on. Oh my God, that explains what I used to see. So my grandparents had a farm, and they raised all their own animals. So every Easter, my grandfather would get 100 chickens. And then by September, when it was starting to get cold, the chicks had all grown up, and we would end up butchering all those chicks, and they would sell it or distribute it all with the family. So we would have these family get-togethers for taking care of all these chickens. And then there was a pecking order as to who did what part. And when you're a kid, you sometimes get the parts that the adults don't want to do. So my older brother and I had to gut the chickens. My grandfather would cut the head off. My older brother and I would gut the chickens. And then my mom and my grandmother and my aunts would pluck the chickens. So it was a similar line process. Well, if you have a chicken that you just killed and you open their guts, their small intestines quiver. And so when I was a kid, I could see this quivering of these small intestines. Oh my God, what? Are there worms in there or something? It's just like, what's going on? Then you learn that smooth muscle is autorhythmic. It doesn't need your brain. So it continued to contract and relax, which made the small intestines quiver. Yeah. Oh, that's why. And it's involuntary again, so we don't have to think all the time. Okay, stomach churn. Okay, stomach churn. So our body takes care of it, so we can think about our anatomy and other items and not have to worry about those basic functions. Yes.
I'm just curious because I know with chickens when you cut their heads up, they can run around for a while.
Yeah.
So how is that possible? Because I'm sure their legs don't have any bones.
Yeah, what actually happens is when the brain is removed, it was controlling normal patterns of skeletal muscle activity. So you go through a period where all the muscles just kind of contract randomly until they run out of energy, then they stop. So yeah, chickens will run around, try to fly without their head for a little bit. So my grandfather used to put a long rope up, and then he would stick the chickens, the chicken feet in a noose, and then cut their head off, and that way they would flap on the rope and wouldn't run around because they damaged themselves. So yeah, if you went back to the last election, and there was an advertisement done with Sarah Palin, and in the background they were sticking turkeys down in this funnel, and the guy was cutting heads off, and the turkeys were. I thought that was the amazing thing that they put on the TV as an ad for an election. So, yeah, if you read it, different people have comfort zones. So, but if you went back and read some of the stories that were written in France when France killed people with guillotines, sometimes the bodies would sit up and move off of the table without their heads. So we actually do the
So
same.
I didn't know that.
We actually do the same thing as chickens.
And I heard that they used to make wagers on how long it would take a body to.
Yeah. Yeah, if you read some of the stories, I mean, in today's world, we would be like, I can't believe they did that. Yeah.
They asked people before they would die. Stories they asked them before they died. In the name of science, if you're still alive after we cut your head off, can you blink a few times and stick your tongue out?
Now, there were actually physicians who would be there if they were doing a number of people in a row, and they would take the baskets of heads back to their labs and actually do some experiments on how long it is. Anyway, that's the gross side of it. So smooth muscle is really slow contraction. And that's because we use it for things we don't need real rapid contractions for, like moving things through our digestive system. So we don't need to do that in a rapid way. The birthing process with the uterus is smooth muscle. So we get these long, slow, sustained contractions. Can you imagine what the birthing process would be like if smooth muscle did this? You'd shoot the baby across the room. Yeah. So very slow contractions, right? And probably good. But if you think about it, when we don't have anything in our digestive system, we don't need all this contractibility. But if we put stuff in our digestive system, then we need contractibility. So we actually do have to control it with two different neurotransmitters. So we have our two buddies that we just talked about in our cardiac muscle, acetylcholine and norepinephrine. But what happens is that if you're really active physically, like trying to swim or run or do something, then you want to move blood from other parts of your body to your skeletal muscles so you can feed oxygen to it. So you would actually want to slow your digestive system down so you can move blood away from your digestive system to actually when you're exercising. So the interesting thing that happens is the acetylcholine has different receptors on skeletal muscle versus cardiac muscle. And so it has a different effect. So on skeletal muscle, if we introduce acetylcholine, and on the three types. So if we introduce acetylcholine to skeletal muscle, it makes it contract. If we introduce acetylcholine to smooth muscle, it makes it contract. So those receptors are the same. Cardiac muscle has unique receptors. So if you add acetylcholine to the heart, it slows its contractibility down. Okay? So the epinephrine or norepinephrine wasn't we didn't talk about it in skeletal muscle. We only talked about cardiac muscle. But it was the one that accelerates cardiac muscle activity. But it actually slows smooth muscle activity down. So they inverse their roles in smooth muscle compared to cardiac muscle. That allows you to change what you're doing in your body so that if you do need increased cardiac activity because you got all this skeletal muscle activity going on, then you can slow smooth muscle activity down and accelerate cardiac muscle and manage all the oxygen that you need to manage that and blood flow patterns. So it's really kind of cool. And then actually the cool thing is that smooth muscle has considerable regeneration. So that's really where a lot of focus has been on looking at trying to get cardiac muscle to repair itself, is the fact that smooth muscle has some stem cells we call spherocytes, and they're much more active than the satellite cells of skeletal muscle in terms of their mitotic abilities. So they've actually been looking at using smooth muscle to try to help regenerate cardiac muscle. And they've got limited success with animals at Johns Hopkins University. So when we look at smooth muscle, it's not striated because it doesn't even use actin and myosin. So cardiac muscle and skeletal muscle use actin and myosin as the contractile proteins. Smooth muscle actually is constructed exactly like we talked about the Y cells have their shape. The smooth muscle actually has an internal skeleton, a cytoskeleton. And the cytoskeleton connects to the membrane. And then the cytoskeleton in smooth muscle is made up of intermediate filaments that allow the cell to shorten or elongate. And so when you see a relaxed smooth muscle, these fibers are longer. And then what you see in the contracted smooth muscle is the fibers have all shortened. And then that's what actually pulls the cell itself toward the center to make it contract. So kind of very different pattern by which it's put together. So what connects the intermediate fibers together are these dense bodies. So actually what happens is the intermediate fibers shorten between the dense bodies, and that's what causes the contraction to occur. But they're small cells, and they aren't moving a big distance like a skeletal muscle cell has to shorten quite a bit of distance to do this. Whereas smooth muscle cells shorten a little tiny bit, but you got thousands of them all shortened. So you can get a distance of shortening by having a bunch of those tiny cells. Where skeletal muscle is shortening from a tendon of origin to the tendon of insertion itself. So they're constructed to have a greater contractibility. It's quite interesting. And what I was talking about is if you looked at the digestive system, visceral smooth muscle, you have cells with gap junctions. So then you only need a couple of nerves going to the muscle, and then all these cells act as a unit because they have gap junctions. So that's like cardiac muscle. And then this is like smooth muscle, where every cell has a connection to the nervous system. And the nervous system has to control every cell in the muscle tissue. And so this multi-unit smooth muscle is just like skeletal muscle. So when we look at smooth muscle, it shares characteristics with both cardiac and smooth, depending on where you find it in your body. So in lab, when we look at smooth muscle, what we'll do is we'll probably look at the digestive system wall. And what we have, because we need to move things through our digestive system, is we have muscle that goes around the digestive system like this and can kind of close it off. Then we have muscles that run the length of the digestive system, and they can shorten to move things along. And so we have two unique orientations of the muscle itself. And so this is classic smooth muscle, where we have a single nuclei per cell, long, kind of tapering cells. So remember slide 19, we looked at those long, thin cells. That's classic smooth muscle. And so in one place in the digestive system, in one layer in the digestive system, you're going to see the cells with the long axis. The nuclei are going to be elongated on the long axis. And then if you move out to the next layer of smooth muscle, it's actually going to look like this. And that's because you're no longer looking at the long axis of the cell like this, but you're looking at the end of the cell. And then the nuclei are perfectly round. So what we have in our digestive system are muscles perpendicular to one another. So when we cut through, we get two kind of unique layering patterns. So this would be what we would see when we look at the digestive system. And so this would be the inner layer of muscle that we would see in the digestive system wall. We call that the circular layer because the fibers are going around the lumen like this. And then the outer layer of muscle we call the longitudinal layer because the cells are running the length of the small intestine. So when you cut the small intestine, you get those two perpendicular views of the cells themselves. So these cells are running this way, these cells are running like that as you're looking at them, which makes it look very different from the two. And then this was the one I was talking about. Remember the erector pili muscle that's connected here, the smooth muscle as well. Okay, so what we're going to do is we're going to talk about for how muscle works. We're going to use skeletal muscle as our model in this unit. And then the differences that we see in cardiac muscle and smooth muscle, we're going to wait until we get to the cardiovascular system to talk about it. And so to try to make it so that you don't have to know all the differences in the physiology of the three muscle types now, I want you to focus on how skeletal muscle works. And then later we'll refine that for the other muscle types. That way you won't get confused as easily with it. Okay? So what I did is I set up a sequence of events that would have to happen for you to actually be able to contract a muscle and relax a muscle. Okay? So to go through one contraction and relaxation. And so what I'm going to do is just read through them real quickly. And then what we're going to do is go back one at a time, and then we're going to talk about details. Okay? So this is like the story, and then we're going to go back and fill in the background for the story. So because skeletal muscle is voluntary, then your brain has to send a signal. And so the signals coming from the brain are actually electrical stimuli created on membranes. And we can actually measure them on the surface of your skin, the electrical stimulus, using instruments, all right? So that electrical stimulus, when we're talking about a nerve, is called an action potential. So nerves that go to skeletal muscle and make muscle contract are called motor nerves. Okay? So what we have is an action potential on a motor nerve that stimulates the skeletal muscle at the junction between the nerve and the muscle, which is called a neuromuscular junction. Okay? Now what's going to happen is we've got channels in the membrane of the muscle cell. And what we're going to be able to do is use the electrical stimulus to open and close channels. And if we open these channels, they're like a window, and certain things will fit through the window and they can move. So the first thing that's going to happen is we're going to open a sodium channel. And then because sodium is in different concentration inside and outside the cell, then sodium is going to diffuse. And when we use the word diffusion, we always have to understand that things move from an area of high concentration to an area of low concentration. So what the sodium is going to do is diffuse. It's going to diffuse in a very predictable way, going from an area of high concentration to low concentration. But it just so happens that sodium is higher outside the cell than inside the cell. So when we open these sodium channels, we're going to move sodium across the membrane. Because sodium is a positively charged ion, then what we're going to do is change the voltage on the membrane by moving a positively charged ion. So what we have is a word that we use for the change in voltage in one direction, and then we use a different word for the change in voltage back the other direction. So depolarization will cause a change in voltage in one direction. It's the shape of the myosin head and pulls the actin toward the center of the sarcolemma. And so we create these crossovers. We're going to use ATP, break it down into ATP plus phosphate. The energy released is going to create a power stroke. And then the power stroke is actually going to pull the active molecules toward the center. So as all the active molecules get pulled toward the center, the sarcomere shortens. Every sarcomere in a myofibril does the same thing. Every myofibril in the muscle does the same thing, so the cell physically shortens. That is some cool stuff. So, why, if you want to lose weight, is exercise a good thing? Because every muscle contraction uses a GABA ATP to make these power strokes. All right. So to reverse that, so that our muscle doesn't stay contracted, we've got to change the charge on the membrane again. So since the first one was a depolarization, the second one is going to go back to where it started, which is going to be a repolarization. So the depolarization takes the membrane in one direction, repolarization takes it back the other direction. So what we have to do is we have to repolarize the membrane. But because sodium was the ion that you moved first, we've already messed up the sodium, where the sodium concentration across the membrane. So we're going to use a different ion in repolarization than we use for depolarization. And it's going to be potassium. And if you look at the periodic table, sodium is smaller than potassium because it's in row 3, where potassium is in row 4. So we can have channels that are unique to those ions because of the difference in their size. And so now potassium is going to rush out of the cell and create this repolarizing event, which is going to cause the sarcoplasmic reticulum to repolarize. And then we're going to collect all of our calcium again. And when calcium comes off of troponin, the molecule returns to its original shape, and after myosin no longer interacts. Now, so now what we're going to do is we're going to do the detail of this. Okay. So Monday in lab, what I'm going to use is a physiograph that uses electricity, and what I can do is put some electrodes on your arms, and I can actually make your muscles contract. and we can think about this whole process and use electricity to do it. So it's similar to what you would do. It's similar to what you would do in a PT's office if you've got a muscle and they have the TENS unit on, they make your muscles, or the app thing you can buy on the TV to give you a six-pack where it's supposed to go around your waist, make your muscles contract all the time. You really didn't need to do exercise to do it. But we're going to use a similar machine, okay? So what I've done in the rest of this is I've highlighted one thing, and then here's the detail for it. So then I've highlighted the next thing, and then here's the detail behind that. And then I've highlighted the next couple things,